Hello students, today we will study defense mechanisms found in plants. There are different kind of defense mechanisms are present in the plants which prevent plants from the infection from the pathogen or defend themselves after the infection of the pathogens. These defense mechanisms can be classified into two categories: structural defense mechanisms as well as biochemical defense mechanisms. Again, further, these two kinds can be classified: pre-existing structural defense mechanisms and post-infectional or induced structural defenses. Biochemical defenses similarly, pre-existing biochemical defenses and post-infected biochemicals or induced biochemical defenses. Defense means to get rid of from the pathogens. Structural defense mechanisms: the surface of the plant or host in first line is the first line of defense against the pathogen. The pathogen must adhere to the surface of the host to penetrate it if it is to cause infection. If it remains unable to adhere, it is enter the pathogen inside the host. Structural defense mechanisms are mainly of two types, as I told you in the flow chart: pre-existing and post infected. Pre-existing structural defenses can be classified into some categories according to the structure or chemicals deposited on the surface of the adherence: wax, thick cuticle, thickness and toughness of the outer wall of epidermal cells, stomata, sclerenchyma cells, and the lenticels are the pre-existing structural defense mechanisms found in the plants. Induced structural defense mechanisms: cellular defense structures, hyphal sheathing. Second is the histological defense mechanisms or structures: formation of cork layer, formation of abscission layer, formation of tylosis, and the deposition of the gums. First, we will study the pre-existing structural defenses. It includes amount and quality of the wax and the cuticle, shape, size, and location of the natural openings, that is stomata and lenticels, presence of thick-walled cells in the tissues of the plant and hinder the advance of the pathogens. Here we can see in the diagram the of the this is the section cutting of a leaf. It is the mixture of long chain. It is coated by the wax. Wax is the mixture of a long chain, a non-polar lipid, it forming a protective coating on plant leaves and fruits, synthesized by the epidermis, excreted by extremely hydrophobic or do not allow water to adhere itself. It's on its surface. Cuticle. This is the cross section of a typical dicot leaf showing cuticle. Disease resistance in Berberry species infected with Puccinia graminis tritici has been attributed to the tough outer epidermal cells with a thick cuticle. In linseed, cuticle act as barrier against Melamsora linea. Silicification and lignification of epidermal cell offers protection against Pericularia oryzae and Streptomyces scabies in paddy and potato respectively. Thus, cuticle is helpful for the host and defend the host from some pathogens in some way. Sclerenchymatous cells. Sclerenchymatous cells, as we all know, is made up of lignin, a thickened walled tissues. Sclerenchyma cells is present in stem and leaf veins. Brittle cells help in mechanical support of the plant, effectively block the spread of some fungal and bacterial pathogens that cause angular leaf spot disease. Similarly, with there are many natural openings. Some of these are stomata. Most of pathogen enter plants through the natural openings stomata. Some pathogen like rust of wheat, Puccinia graminis species triticide can enter its host only when the stomata are open. Structure of stomata provides resistance to penetration by certain plant pathogenic bacteria. This is the diagram showing structure of the stomata, opening and closing of the stomata. And now it is lenticels. Lenticels are opening on fruits, stem and tubers that are filled with loosely connected cells that allow the passage of the air. Here you can see lenticels on apple. Here these are the lenticels on the stem potato. Shape and internal structure of lenticels can increase or decrease the incidence of the fruit diseases, stem diseases, etc. Small and suberized lenticels will offer resistance to potato scab pathogen Streptomyces scabies. Thus, lenticels also help in defense mechanisms. Now we will study the post-infectional or induced structural defense mechanisms found in the plants. Most pathogens manage to penetrate their host through wounds and natural openings and to produce various degree of infection, as we had studied earlier in the pathogenesis topic. Pathogen penetration through the host surface induce the structural defense mechanisms in the host cells. These may be regarded as histological defense barriers, core clear. Histological defense barriers include core clears, abscission layers, and tylosis formations. As you know, core clears and tylosis, these all are formed during secondary growth of the plants in dicots. Cellular defense structures, hyphal sheathing on the host surface, can defend host from the infection. Histological defense structures, core clear infection by fungi, bacteria, and some viruses, nematodes include plants to form several layers of cork cells beyond the point of infection. These cork cells inhibits the further invasion by the pathogen beyond the initial lesion and also block the spread of toxin substances secreted by the pathogen. It also stops the flow of nutrients and water from the healthy to the infected area and deprive the pathogen of nourishment. Thus, cochlears are very beneficial to the host as defense histological structures. Potato tubers infected by Rhizoctinia prunus domestica leaves are attacked by Cocomyces. Here you can see the lenticels cochlears. It is infected by the pathogen, but cochlears are not allowing further invasion of the pathogen to the host. Abscission layers. An abscission layer consists of a gap formed between infected and healthy cells of leaves surrounding the locus of infection. Due to the disintegration of middle lamella of parenchymatous tissue, how abscission layer forms? It forms due to the disintegration of the middle lamella of parenchymatous tissues. Gradually, infected area shrivels, dies, and slugs off, carrying within the pathogen. Abscission layers are formed on young active leaves of stone fruits infected by fungi, bacteria, or viruses. This is the diagram showing the formation of abscission layer in this section of leaf, and it stops further infection, spreading of the further infection in the plants. Tylosis. Tylosis are the outgrowths of the protoplast of adjacent living parenchymatous cells which protrude into xylem vessels through pits. Uh, these are the diagrams of the tylosis. Here you can see balloon shaped cells which, which are filling the xylem vessels. These are known as the tylosis. Tylosis have cellulosic cell walls. It is formed quickly ahead of the pathogen and may clog the xylem vessel. As pathogen arrive in the cavity of the xylem, they form tylosis and clog the xylem vessels, completely blocking the further spread or further advancement of the pathogen in resistant varieties. Tylosis, diagram of the tylosis. Gum deposition. Various types of gums are produced by plants. Many plants produce gums around lesions. Lesions are the spores produced by the pathogen after the infection or after the injury are known as lesions. And the various types of gums are produced by many plants around the lesions. Gum secretion is most common in stone fruit trees but occurs in most plants. It is common in stone fruits. Generally, these gums are exudated by plants under stressed conditions. When they exudate during stress conditions. Gummosis is the process in which gum produced by the plants and trees. So now we will study the cellular defense structures. This diagram is showing hyphal sheathing is observed in flax infected with Fusarium oxysporum. Hyphal sheathing, diagram showing hyphal sheathing. Biochemical defense mechanisms. These are also pre-existing and post-infective defense mechanisms. Pre-existing defense mechanisms involve inhibitors, phenols. Inhibitors can be released by plant in its environment or present in plant cells before infection. Second is the phenols. We have already studied the phenolic compounds as the defense mechanisms or biochemical defense mechanisms. Induced chemical defense substances, hypersensitive response, production of antimicrobial substances, phytoalexins, and plantibodies. Phytoalexins we had already studied. Pre-existing chemical defense. Although structural characteristics may provide a plant with various degree of defense against attacking the pathogens, it is clear that the resistance of a plant against pathogen attack depends not so much on its structural barrier as on the substances produced in the cell before or after infection. Before infection or penetration of pathogen, host release some chemicals to defend themselves. Inhibitors released by the plants in its environment. Plants exude a variety of substances through the surface of their above ground parts as well as through their underground parts, the roots. Inhibitors released by the upper ground parts and the underground parts of the plants. Inhibitory substances directly affect microorganisms or encourage certain groups to dominate the environment, which may act as antagonistic to the pathogen. What is the antagonism? There are there is a mechanism by which a living organism can inhibit the growth of the other living organism is known as the antagonism. Root exudates of the there are two examples. Root exudates of marigolds contain a tertinyl, which is inhibitory to nematodes. Similarly, Cicer aratinum chickpea resistant varieties have more glandular hairs which have malic acid, which inhibit spore germination. This diagram is showing production of toxic compounds in rhizosphere or phyllosphere. Inhibitors present in plant cells before infection. It is becoming increasingly apparent that some plants are resistant to disease caused by certain pathogens of an inhibitory compound present in the cell before infection. It is stored in vacuoles of plant cells. Phenols we have studied already. Phytoalexins we had studied already. Hypersensitive response we have to study. The term hypersensitive uh, it, we will study it in our next class because we will unable to upload the bigger video on the YouTube. Thank you.